To start with, this is part of my PhD project, which, as Larry said, I'm about to finish. Just to give you an outline, I'm just going to very briefly discuss coral reef protection and connectivity along the southwestern Indian Ocean, try to define quickly ecological genetic connectivity, then with that in mind, explain some of the two concerns that I have found for reef conservation success in South Africa, then the results of my assessment of ecological genetic connectivity, and then discuss and provide some management implications and recommendations. So very quickly, what I did, I defined the southwestern Indian Ocean. I include only Mozambique and South Africa, and those are some of the protected areas that we have there. About 5% of the reef area is protected. It's WWF is trying to, I think it's by 2016, trying to come up with a network of MPAS, which are assumed to be connected thanks to the oceanographic patterns of this region. With that assumption, many studies have been done on the genetic connectivity. Those are some of the ones that have been done on corals, and I'm only showing the ones on coral, not fish, because I believe that corals or sessile organisms, no mobile organisms, will show you true connectivity. Fish will show you connectivity not because of gene flow, but because of movement of adults, but that's another discussion. But anyway, those are two species that have been worked already for genetic connectivity, and using different markers, the locations, the scale of the project, the FST, which is an indicator of genetic variation, and what they found, if they were connected, all those reefs, and the source at the end. The starts for the FSTs, which is the genetic variation, or a measure of genetic differentiation. As you can see, it shows that for some, for Acropora, for Platygyra, and Poecilophora verrucosa, for some specific markers, they show significant genetic variation. In other terms, it seems like there is there is no connectivity or not enough gene flow. However, those that there is some study that found panmixia. Panmixia means that they are all connected, and those are just by markers like the ITS and the allozymes, which are have a low mutation rate. So they give you what happens many years ago. When you assess or use markers like microcytes or nuclear introns, we give you a snapshot. They show you that there is no connection. There is not sufficient gene flow. Those markers allow you to measure genetic connectivity, which or ecological genetic connectivity, which in an extreme concept, which about closed and open populations and the risks depending on ecological factors is very relevant. Ecological genetic connectivity, what I define is that happens at short-term spatial scales that will give you information about the resilience of a reef, or the capacity to replenish the capacity of a reef to replenish itself, or from larvae from other reefs. That's some of the potentials of how to measure the extremes for ecological genetic connectivity. And this ecological genetic connectivity is of high concern to managers, because as it happened, recruitment, mortality, and so on, those are factors that happen at time scales that concern managers, or that managers can or are mandated to deal with. So in the South African case, I'm not going to go further here, but we have the Maputaland Reef, that marine protected area, which has been divided in 3 complexes, and that area, the ones on the north are sanctuary zones, the one at the south, the southern complex, are sanctuary zones as well. And the central area is the one that is heavily used. Tumal Reef specifically is the most heavily used reef. All that area is managed under the assumption that if we protect the ones on the south and the ones in the north, it's sufficient enough, because of the flow of the current would allow that reef from the north, that the larvae from the reefs to the north will disperse and then replenish those reefs to the south. However, with that information that I showed you at the beginning about the genetic connectivity, there is some questions about this assumption of if there is actually that north to south flow of any larvae, if at all. So that brings to these two main concerns for reef conservation, which is are individual reefs isolated at ecologically relevant spatial and temporal scales, and is the current protection approach appropriate for the degree of connectivity that we found or that we see in our reefs. So to do that, I chose two species. As I said before, I chose hard corals because I believe they give a better picture of the real connectivity. And these two species they have the same reproductive strategy. They are broadcast spawners. So what is expected? Palmixia as well. They are supposed to be connected because of the flow of the current. However, they have two different characteristics. One, what I call the rabbit, is Acropora austera that Mike just mentioned, and then the tortoise, which is Platyjara, because of the growth rate. Acropora grows very fast, Platyjara is very slow. As well, the settlement is different. Platyjara it settles very quickly, so its potential to limit the dispersal is very high, because it can settle as soon as it fertilizes, 2 days after fertilization, whereas Acropora austera can take a little bit longer. And as well, in terms of management and the way that it can spread that species, Acropora austera is very susceptible to breakage, so there can be fragmentation and asexual reproduction. And Platyjara, since it's a massive coral, it doesn't have that. In terms of climate change, two different characteristics. Acropora austera, very weak. Platyjara daedalea, very strong in terms of temperature resist tolerance. So with this in mind, I try to assess the genetic connectivity to have in a Palmixia hypothesis, two species that can have different characteristics for climate change. What picture are we going to get? So I address the fine scale. Sorry. Yes, I address two scales of connectivity. The fine scale genetic connectivity, which I took Tumor Reef. I chose Tumor Reef to sample there. I divided the reef in several areas. This is Tumor Reef here in the offshore area and the inshore area. 8 sites. Northern, central, south central, and south sites. And on each of those sites, I laid a transect, and I sampled every colony that I found 2 meters apart, 2 meters along, 100 meters transect. So I managed to map as well each coordinate, obtain abundance and density data, and size as well. And then I had the large scale connectivity, which I pretty much obtained samples from the other reefs just to have an idea of the bigger picture. How did I went about measuring ecological genetic connectivity? I'm not going to go into genetics here. You can talk to me later, but it's very basic. A genotype, 9 nuclear markers. Nuclear markers because they are hypervariable, and they can give me that short-term information, what is happening for few generations, or few generations ago, in Acropora austera and 7 in Platyjara daedalea. Here I use 2 types of markers, microsatellites and nuclear introns, just to see how much information I can have. When I say genotype, I pretty much screen for each of those markers, establish what I can call a barcode for every single colony that I have sampled. So I had one unique identifier in terms of genetics for every single colony. Then I address, for this talk, I'm going to address only the private allelic richness as an index of how distinct a population is from another. So the higher number of private alleles that are exclusive to me or to this population, it means that I'm not very related to any other populations. And as well, I use an analysis of genetic variation and network theory as a proxy for genetic differentiation within and between reefs, and to estimate gene flow. So this is some of the results. Here, Acropora austera, Platyjara daedalea, Tumor Reef, the fine scale genetic connectivity, and the large scale connectivity here, all the reefs. In blue is the offshore sites for Tumor Reef, in green, the northern the inshore sites. And each of these is, all reefs that I sample additional for the large scale, Letchman Shoal, Red Sands, 2-mile reef, 5-mile reef, 9-mile reef, Rabbit Rock. So we sampled all the reefs in South Africa, and then we sampled some in Mozambique, some Tanzania, Chagos for Platillata, Basaruto, and Reunion Island for Acropora austera. Just here, to give you an idea in terms of 2-mile reef of these both species, pretty much the same number of private alleles. We just show us that even though there is it's a smaller scale, the sites happen to have unique alleles. So there is some kind of separation at that scale, okay? Because each site, or if I can call it here, the samples from that site happen to have a unique genetic information. For Platillata, daedalea, same as well, and you can see how there is a differentiation here between the northern and the south in the reef. So even at that scale, I can start seeing some differences there. When I did an isolation by distance analysis, I did find the distance between the farther apart, the less related they are. In terms of private alleles for the large scale, just to bring you here, look at the break point between Mozambique and South Africa. 9-mile reef and Rabbit Rock are higher compared to the nearest Mozambique reef. So what Mike said about that break between South Africa and Mozambique, we can see it in the number of private alleles. So there is definitely some break point at this area over here. And Letchman Shoal, which is the farthest or the southernmost reef as well, high number of alleles indicated differentiation of private alleles, indicating isolation from the rest or differentiation. Then I went to look at the genetic variation, and here, using a pop graph analysis, what I did is found how the genetic information varies between sites. If there was panmixia at the finest scale genetic connectivity, which is the reef, I will expect all the dots to be connected. But what I found is that actually there is some disconnection between or within the reef, suggesting that there is either two populations. This reef is the northern offshore, which is here, northern inshore, and it goes there, and it goes there. So this appears to be one area, and this appear to be another area. The size of the dot there is related to the density of that side, the density of Acropora austera. So here we show a higher density, few density, and a lower density of Acropora austera. So just give us an idea that there is a breakage here, and that the density is not related to genetic connectivity. You will expect, since this is very dense, that it will spread everywhere, but that is not the case. And in terms of statistic genetic variation, clear, it was significant, suggesting that definitely there is a differentiation at the final scale genetic connectivity for this reef. Now, I use, you guys heard the talk previous, which was the Bayesian clustering analysis of probabilities. So I just look at the genetic information without prior information on the location of those samples. I just put it in the model, and I ask the model, tell me what are the most probable number of populations or clusters or groups on 2-mile reef. These are my 8 sites over here. And just to show you here, it gave me 2. White is higher probability of all of being one cluster. Red is another one. So it divided again 2-mile reef without prior knowledge of the location of the samples, that there is a break on 2-mile reef on this area over there. When I look then, since I had the coordinates for every single colony, I went and asked myself, are colonies that are closer to each other more genetically related? So I ran a spatial autocorrelation analysis to see if the closer the colony, the higher the genetic relatedness. And what I found for Acropora austera, that yes, indeed, the closer they are, the more related they are. If they are less than 10 meters apart, the genetic relationship is outside what will be expected by chance, which is the area within the red lines. And here as well, it's another way of putting it, and while the distance increases, so does the distance increases between colonies, the relationship between, or relatedness between colonies decreases. So that just gives me that there is actually limited dispersal or some aggregation genetic of some kind. For Platygyra, same, a break as well. Densities, you can see how dense this species is on the reef, and funny enough, that density apparently helps to make the reef not genetically different, or the sites not genetically different. So even though the model here found some genetic differences, or split the sites in two, those are not genetically or statistically significant. When I did the spatial autocorrelation analysis for this species, didn't find anything, so it means that the patch size is much larger, and that's why I didn't find any differences in the genetic information between sites. For the large scale, interesting here, I did the same for the reefs. These, I group all non-South African reefs, and you can see how, even though they're closer to each other, they don't appear to be connected. So what would you expect if there was a north to south connectivity by the currents, that every single reef that is adjacent to the next one will be connected? And what I found is actually that's not the case. Reefs that are actually closer to each other, they are not connected, but there is a common source. From Mozambique, it's a spread to other reefs. From Rabbit Rock, it's a spread to other reefs, but there is no connectivity between reefs that are closer to each other. That's for Acropost, and the differences between reefs was significant as well. For Platygyra, found pretty much the same, although there was more connectivity between reefs that are close to each other. So same as the fine scale, I'm finding at the large scale for both species. Closer they are, not necessarily the more related they are in terms of space, but at the individual level, at Tumal Reef, I'm finding that the closer we are, the more related we can be within a reef, but not between reefs. And it was significant as well. So what does it mean for the first concern of if are the individuals reef isolated at ecological, relevant, spatial, and temporal scales? First, well, I found significant genetic subdivision within and between reefs in Acropora austera, significant genetic subdivision between reefs in Platygyra, and a patch size of 40 meters for Acropora austera, suggesting limited dispersal. As well, that northern reefs are not necessarily the source of coral larvae. When I said northern reefs is the immediately next to it, or adjacent reef, and Rabbit Rock and Normal Reef are definitely some kind of break point in genetic connectivity for these reefs. All of that suggests signs of isolations, and that will be one of the first concerns for conservation and for the management of these areas. When I put it into context, this is my study, weekly, using microsatellites and nuclear interns for Acropora, for Platygyra as well. So that concept that our reefs are not well connected has actually increased its weight, which had some management implication. First, to suggest that genetic structure is not a simple function of reproductive strategy. If we are broadcast spanning, not necessarily means that we're going to be well connected, that reef site proximity is not related to connectivity, that there are some microhabitat conditions that at the finest scale will make two sites not related, that a blanket approach for reef management is questionable, and that isolation in general will diminish replenishment of corals community from nearby healthy corals and may limit recovery, that isolation that we observe, which suggests that that approach needs revision. What can we suggest? And just to finish, maybe definitely reinforce the protection of sanctuary zones, because whether they are not connected, there will still be in the large temporal scale, there are going to be sources of larvae, protection of adult colonies to ensure adequate supply of recruits from the adjacent reef, protect sites within a reef rather than the whole reef, or establish within reef conservation management units, in the case of Tumba Reef, preferably situated on the north side. Some acknowledgements, and thank you very much. Sorry if I went very quickly.
Thanks, Phanor. I just want to check, is Gillian here? Ah, okay. One question then, Phanor. Anyone got a question for Bruce?
Thanks very much, Phanor. That was very interesting. Just, it might be obvious to you, but it wasn't to me. What is the larval duration from fertilization to settlement? Because that obviously will determine how far the spawn are going to...
It's called the competency period after fertilization. Before fertilization is how the larval, it can last, I think they can fertilize a couple of hours after they have spawned. The larvae then can stay in the water for up to 120 days. Okay? So the potential for dispersal is humongous. But it's not the case what we're seeing. What my hypothesis, and this is for the discussion of my PhD, is either we finding definitely isolation and limited dispersal, and then I will relate that to size measurements that I have to see if size and age measurement to see if the older colonies are the potential parents of the younger colonies, so to suggest limited dispersal. And the other one is that actually kind of a settlement or replenishment in batches. And so it's a spawn somewhere in the north, it comes, find somewhere to settle, and since they have all the common source, they will settle. And that's why they happen to be all related. So they just have a common origin and they settle all together because it's suitable for them. But yeah, the capacity for dispersal is there, they have it.
Thanks, Phanor.